Welcome to week 4, where we're going to talk about how we take DNA and make a protein from the information in the DNA. So today is all about transcription and translation. We're going to be drawing, so get yourself a pen and some paper and get ready to draw with me. Okay, let's start with our overview of our goal here. So we're going to take the information in DNA and we're going to use that to make a protein. That's our goal. What's actually going to happen is our DNA contains pieces of genetic information called genes. Genes have the information we need to make a protein, except the genes are part of the DNA, and the DNA cannot leave the nucleus. So we have to make a copy of the gene in a molecule called mRNA. The M stands for messenger. It's a messenger because mRNA can leave the nucleus, so this is stuck in the nucleus. mRNA can leave the nucleus, and it can go over to the ribosome. And the ribosome is the thing that actually makes your protein. So that's our goal today, is to make a protein. Let's draw it out, because we are fabulous artists and we love drawing. This is my plasma membrane. Little lopsided, that's okay, it's still beautiful. Inside my plasma membrane I have the liquid, which is the cytosol. I'm going to have a Golgi apparatus, and I'm going to have endoplasmic reticulum and mitochondria, all of the goodies, the lysosome, vesicles, all of the cool stuff that's inside my cell. But we're not going to focus on that today. Today we're interested in talking about what's going on inside the nucleus. So I made my nucleus big. There's my nucleus. This is the nuclear envelope. There are little holes in the nuclear envelope. Those are called nuclear pores. So however you want to draw a hole. Little holes where things can go inside and outside of the nucleus, nuclear pore. Our mRNA is going to sneak out from a nuclear pore in a little bit. And then inside my nucleus, I have my DNA double helix. So there it is, beautiful double helix. We know all about the structure of that from our first chapter, and we know that we think Rosalind Franklin is super cool because of it. Now I'm going to take this DNA double helix and I'm going to open it up, and then we're going to look at one piece of it. This piece of the DNA that we're going to look at, we're going to call a gene. And it's called a gene because this piece of DNA is going to get used to code for a protein. We're only going to look at one half of the DNA, so the other half of the double helix is over here. And let's say that this DNA has the sequence T, A, C, A, G, G, T, C, C, A, T, T. Just for funsies. So I've got my sequence of DNA. What I need to do now is I need to make a copy of this gene that can leave the nucleus. My DNA is too big to go anywhere. My gene's a piece of the DNA, so it doesn't get to hop off. It has to stay part of the DNA, so it's stuck in the nucleus. And the way I'm going to make this copy is by making a piece of mRNA. So this is going to be my strand of mRNA, and my strand of mRNA is going to be complementary paired with my gene. So what pairs with T? It's A. What pairs with A? It's T, except this is RNA. Remember, RNA uses uracil instead of thymine. So there are no T's, instead it's a U. What pairs with C? And then A pairs with T, G pairs with C. Oh, I ruined it. What does A pair with in RNA? U. See, it's tricky. You gotta pay attention. G pairs with C. This G would pair with this C. This T would pair with this A. I've got a G, a G. A pairs with U. I won't make the same mistake twice. And then A. So this is my piece of mRNA. It is a complementary pair with my gene. So it matches my gene. It would base pair right there. And my mRNA is allowed to leave the nucleus. And I needed to make a copy of the gene in mRNA so I could take it to the ribosome, which is going to be outside in the cytoplasm. Now this mRNA is called mature once it's got some modifiers added to it. Right now it's immature. Our modifiers are things like a cap. The cap prevents your mRNA from being degraded. I like to think of it kind of like a hard hat. So you're putting a hard hat onto your mRNA to protect it. We're also going to add a tail. So you get a hat and a tail, or a cap and a tail. And the tail is called a poly A tail. And that's because the tail is actually made of a whole bunch of repeating adenines. So just a whole bunch of A's. That's what poly means, is many. So poly A tail added to the end of my mRNA. And another thing I'm going to do to my mRNA to make it mature is I'm going to remove the introns. So I didn't draw any introns in here because I didn't want to make it super long and cumbersome. But introns are pieces of the gene that are not used to make a protein, but are probably used in gene regulation. So we can cut those out before we take our mRNA to our ribosome because we don't need them to make the protein. I remember that we remove the introns because I think introns are interruptions. So introns are interruptions. We want to get rid of the interruptions so we have just the information we need to make a protein. My mature mRNA can now leave my nucleus through one of the nuclear pores, which is a hole in the nuclear envelope, and my mature mRNA will find its way over to a ribosome. A ribosome has two parts. Ribosome. It's got a large subunit, which is like the top of it's bigger, and then it has a small subunit. The smaller part on the bottom. I think it kind of looks like a short squat mushroom. So there's your short and squat mushroom. What the ribosome is going to do is the ribosome is going to take amino acids that are floating in the cytoplasm. I'm going to draw them as little pearls. And it's going to take these amino acids, they're going to go toward the ribosome, and it's going to connect them together into a chain. So it's taking the monomers of a protein, amino acids, and it's connecting them into a polypeptide. This is the primary structure of the protein, the order of the amino acids. These amino acids have charges on them, and so they'll fold up and they'll create the secondary, tertiary, and quaternary structure, if there is a quaternary structure, for your protein. So that's the goal of my ribosome, taking these amino acids that are just floating around in my cytoplasm and adding them together into a polypeptide. It's a little bit more complicated than that, so let's draw this process here and talk about how we make that polypeptide. This all happens in the cytoplasm. I'm just going to draw it down here. That's an O. So this is happening in the cytoplasm. Here's how it works. I've got my mRNA. My mRNA is going to have a start and a stop. The start has the same code. The codon for start is always AUG. For some reason, this reminds me of the Goonies, and I think about Sloth saying, "Hey, you guys," and so AUG, that's where we start. The stop codon comes in a couple different varieties. One is UAA, another is UAG, and another is UGA. So there's 3 types of stop codons. And I remember them with an acronym. So each one of these things is saying something mean, and that means stop. We have you go away, you are gross, and you are awful. You go away, you are gross, you are awful. So those are our 3 stop codons. No means no, stop, you are awful. If that helps you remember it. Inside of my mRNA, I'm also going to have other pieces of genetic code that come in sets of 3. We call those our codons still. And each of those pieces is going to code for one specific amino acid. We can figure out how these code for amino acids by using our amino acid chart. Okay, so this is the amino acid chart from our textbook. This is our textbook. This is actually my computer. I'm poking the screen right now. So we can use this universal genetic chart to figure out what our codons are coding for. So in our drawing that we just did, we had a codon of UCC. So we want to use this chart and figure out what amino acid UCC stands for. We go here, we look at the first letter. Our first letter is U. So we're going to be in this row here. Then we look for the second letter. Our second letter was U. So we're going to be in this box. And our last letter was C. So UCC as a codon is going to code for the amino acid PHE. So there's 20 amino acids. They each have a fun name, and each one gets a 3-letter abbreviation. So this will be our first amino acid. If we look back at our paper, our next one was AGG. So first letter A, we're going to be in this column. Second letter G will be in this row, this column, this row, and then G. AGG, that codon stands for ARG as an amino acid, arginine. So note that if you were to change one of the nucleotides, you might still get that amino acid. That would be a silent mutation. So the fact that there's a couple different ways to get these amino acids are how come we can have silent mutations. Then on our amino acid, or our piece of mRNA here, our next codon was CCC. So take a moment, look at this, and find what amino acid does CCC code for. Pro. So pro will be our third amino acid. Then we'll have a stop. You can see your stop codons here. 3 ways of writing stop. And at that point, your protein will break apart from the ribosome and you're done making it. Okay, now we have a piece of mRNA that has the code AUG. That's our start codon. UUC AGG CCC, and then the stop codon UAA. This is our piece of mRNA. When we are ready to make a protein from our mRNA using the ribosome will come and it will sit on top of our piece of mRNA. So here's the small subunit. Little small subunit attaches, and then on top of it, the large subunit. As it attaches, it's going to move down the piece of mRNA, and it's going to move down one codon at a time. So here's its beginning position. Then it scoots down. The large subunit is over the next codon, and then it scoots down. Then it's over the next codon. Then it scoots down. Then it's over the next codon until it gets to the stop codon. When it gets to the stop codon, it sees, "You are awful, oh no!" And so the whole thing kind of just breaks apart, and the large subunit pops off and the small subunit pops off, and they both float away, and that's how you know you're done making your protein, because as soon as they see the stop codon, everyone lets go of the mRNA and just kind of floats apart. So how does this process make a protein? Here's how it works. Inside of my ribosome, the large subunit and the small subunit, I have 3 spaces. These spaces have names. They are E, P, and A, which I remember because of the Environmental Protection Agency. That helps me remember. There's also another way that helps me remember what these places do. The E site is almost always empty. The A site is where you add a new amino acid. I'm going to abbreviate that AA. So empty for E, adding for A, and then the P site is where you hold the polypeptide. That's where the polypeptide sits as it's growing. I have my piece of mRNA. I have my start codon. My start codon will attract a tRNA molecule, and a tRNA molecule will sit on it holding our first amino acid. That's 1, amino acid number 1. So I have my start codon, I have the amino acid, tRNA, I think T for transfer, it's transferring the amino acids over. This is an amino acid. It's attached to the start codon. The first start codon always codes for methionine, so that's got to be a met amino acid. Now my large subunit and my small subunit of my ribosome will sit on my mRNA, and my E site and my A site are empty. So the first tRNA is sitting in the P site. Here's what happens next. I've got my ribosome, I've got my mRNA. What happens next is the first amino acid is sitting there in the P site. The next amino acid is going to be pulled over. Here's amino acid number 2. And it's going to go into the A space because I'm going to add this amino acid. It's going to pop in there, and when it pops in there, the ribosome is going to shift down so that this tRNA gets pushed into the E site, and this amino acid gets added to the top of the new one that was just put into the A site. Ready? This looks like this. So my new amino acid number 2, was added to the A site. Number 1 was in the P site. It's going to shift down. So now I'm going to have this amino acid pop on top, which means I'll have my tRNA now holding 2 amino acids. The A site's empty because this used to be in the A site, but we shifted the ribosome over. The tRNA that was holding amino acid number 1 is now holding nothing. It's sitting in the E site. It will just pop out and it will leave the E site empty. E for empty. So pops out. Now I have a ribosome which has a tRNA holding 2 amino acids attached together, so our growing polypeptide. The E site is empty because the empty tRNA left, and the A site is empty, which is great because that means that I can add my 3rd tRNA into the A site. I'm going to give myself more room. My ribosomes are getting smushed, that's okay. But they look like mushrooms, how exciting. So now I have my tRNA that's holding the first 2 amino acids. I'm going to add the 3rd. So here's the 3rd, it gets added to the A site. My amino acid is going to shift down the mRNA, which means that these 2 amino acids are going to get added onto the top of the 3rd. Try and make this one taller. Large subunit. So now I have a tRNA that has amino acids 1, 2, and 3 sitting on top of it. The tRNA that used to be holding amino acid 1 and 2 is now empty in the E site and it will leave. That means that the A site is open for a 4th amino acid to join. My tRNAs are looking a little squiggly. That's okay. It's still art. We're still beautiful artists. So there's tRNA holding amino acid number 4. And this will continue and continue and continue, creating giant chains of amino acids because we have some really large proteins until your ribosome scoots over and the A site is sitting on top of stop. Once it's sitting on top of stop, a release factor will bind, and the release factor will break apart the big and the small subunit, and they will float away, and you're left with your little mRNA there, and your mRNA will get broken down, and you'll reuse the nucleotides to make another one. When we were looking at our mRNA strand, I gave you a couple codons, right? We had AUG, that's start, then we had UUC, I think we had CCC, whatever it is. The way that these tRNAs know what order to go in is because at the base of the tRNA, they kind of have this gumdroppy shape. There is a anticodon sequence. So at the base here, there's GGG. So this tRNA will only pair with this codon. And so that ensures that the amino acid that's being carried by this tRNA goes in the right place. I don't want to accidentally put this amino acid where this codon is. I only want it to go with the right codon. So this region at the base of a tRNA is called the anticodon region. And so your anticodon is a match to the codon, and that helps ensure that you're putting the right amino acids in order. Otherwise, you could switch up which amino acid goes first, or third, or fifth, and we want to keep the order of our amino acids specific because the order of the amino acids determines the function and shape of the protein. And protein function and shape determines how well your protein works. Remember, if we ruin the shape of a protein, we call it denatured. And if your protein is denatured, it's no longer working, or it's working at a less good quality. It's hurt, for lack of a better word. It's detrimental to denature your proteins. Cool. So, for our overview, inside of our cell, we have our nucleus. Our nucleus contains our DNA. Our DNA has sequences inside of it called genes that code for proteins. DNA can't leave the nucleus, so only mRNA can leave the nucleus. The process of transferring the information from DNA into mRNA is called transcription. You're transcribing the DNA into mRNA, the information in DNA into mRNA. So DNA, mRNA, that's transcription. Now, the information in the mRNA leaves the nucleus and it goes over to the ribosome. That one looks very much like a mushroom. The ribosome is going to take the information in the mRNA and it's going to turn it into a protein. So, mRNA to protein is translation. You're translating the genetic code into a protein. Transcription, translation. Once you've created your polypeptide, it will fold up and it may be ejected out of the cell in a vesicle through exocytosis, if you remember that from 211. Or if it's going to be used right in the cytoplasm, it could just fold up right here and start functioning and doing its job. Okay, thank you for drawing that out with me. I know that was a lot of steps. Feel free to watch this video a couple times or watch it and pause it as you go and draw it, or listen to it and imagine what's happening in your head and then draw it. Test yourself however you want, and please email me with questions. I'll talk to you soon. Have a great day.